Hey everyone, I am Nadja Verspagen and I'm doing my PhD at Helsinki University. And today I will tell you something about my project working with the glandbill fritillary butterfly. So I don't think I have to explain to many of you that climate change is affecting organisms all over the world. And one way they can deal with this is just by moving away from the change, for example, towards the poles or higher up mountains. And this is something that we are already seeing, but not everyone can do it. For example, if you're an organism that can't really disperse easily due to physical limitations or because you're dependent on a food source that's only found in a certain location, for example, then you will have to cope with the change. You can do this in two ways, either by adaptation through evolution, which happens over multiple generations, or through adjustment through phenotypic plasticity, which happens on much shorter timescales within generations. So the extent to which you can do both of these things does determine how well you can survive in a changing environment. And this to me is a really important phenomenon, and I wanted to know more about it. So that's why I started my PhD. So I'm studying these questions by using the glandbill fritillary butterfly as a model system. So one way to study these things is by using environmental gradients, such as latitudinal climbs or altitudinal climbs. And in my research, in my PhD thesis, I use both of them. But in the first chapter that I'm talking to you about today, I'm just using this altitudinal gradient. So what you actually do when you use this method is you take organisms, in my case, these butterflies, from different environments. So from, for example, warm to cold environments. And you can do that by using these clients. And so you take the organisms from different populations and put them under lab conditions where you can manipulate their environment. For example, expose them to stressful hot temperatures. And that way you can separate the genetic effects from plastic ones and thus get a better picture of the overall capabilities of the species across its entire range, how they deal with environmental stress. And this is maybe a bit hard to visualize just in this picture, but it's easier to see using some graphs. So if we, for example, take a population from this lowest altitude, so the warmest environment, and from the coldest environment, the highest altitude, we put them in the lab and we manipulate their rearing temperature. So we just grow them under different temperatures. There can be a few different outcomes. So first of all, it could be that they do respond to the temperature, for example, by growing faster at higher temperatures compared to the low temperatures. But there is no difference between the populations, so no genetic difference. However, it could also be that they still respond to temperature in the same way, but just one population consistently grows faster than the other population. So in that situation, there is a genetic difference, but no genetic difference in the way how they respond to temperature. In this final graph, we again see two different responses. So there is a genetic difference, but also the two populations respond differently to the temperature from each other. So in this case, there is a genetic difference in plasticity, meaning that, for example, this population has a much higher slope than these ones. And so based on some literature, we can actually expect one of these things to happen also in my research. So in this first chapter of my PhD thesis, I aim to find out whether there is a difference in response to high temperatures in the glandbill fritillary butterfly that originate across an altitudinal gradient. So for that, we followed post-diapause larvae all the way from diapause into adulthood. So what we did is we collected these butterflies across this altitudinal gradient at 6 altitudes. At each of these 3, we had 2 families. This one, each of the altitude only had 1 family that we could collect. So we combined them into one group because they're also kind of quite close to each other. So that left us with 4 altitude groups, each with 2 or 3 families each. And those families we separated again over 4 different rearing temperatures ranging from 25 degrees to 34 degrees. And so basically from each family, we took 40 larvae, put them 10 at 25, 10 at 28, and so on. So then we just tracked them through their development, and we measured a lot of life history traits. So first of all, the larval mass over time, the total development time from diapause into adulthood or into pupation. The pupal mass, with that we could calculate the growth rate. When the adults eclosed, we measured their wing size, with which we could calculate the wing loading and the wing aspect ratio. And finally, with the subset of the individuals, we did measurements for the basal and the flight metabolic rate. So because these would be a lot of things to focus on in one presentation, I'm just going to show you the results for the growth rate and the wing loading because I think they are the most important ones. And yes, so here the first graph for the growth rate. So what you see here is the different rearing temperatures and the growth rate on the y-axis. And the different lines are for the different altitude groups. And then we have 2 panels for the females and the males. So the first thing that comes to mind when looking at these graphs is that there's a really clear response to temperature, with larvae at 25 degrees growing much slower than the other ones. And the other ones are actually quite similar in response. And this makes sense because 25 is actually quite a low and stressful temperature for them. So they just really do not like it. What's interesting here, I think, is if we zoom into this a little bit more, and these are the graphs for the pupal mass and the development time. So we basically took the growth rate by dividing this pupal mass by the development time. And here you see that the larvae are not really, like the difference in pupal mass is not really big, and they're actually a little bit bigger at this lowest temperature. However, the development time is really long at 25 degrees compared to the other temperatures. And this shows that maybe the larvae want to reach kind of a high, like a high mass, despite the fact that they're growing at this low temperature. So maybe being big is good for adults. So that's why they still want to reach that temperature, even though it's so cold. So that's why they just grow very slow. The next thing that we can see here is that there's no significant effect of altitude, especially at 25, where they're all the same. But also at the higher temperatures, there's no significant effect of altitude. And this could be maybe because we were very constrained by how many larvae we could collect. So we have a limited amount of families. However, what's interesting is that the order of the altitude seems to make sense. So these ones from the originating from the coldest environments grow the fastest, while the ones originating from the warmest environment grow the slowest. And the other ones are also kind of in the same order. So this shows that maybe with a bigger amount of families or bigger differences between the altitudes, we could have found a significant effect. So even though we didn't find it now, this is kind of quite promising for my further studies with the latitudinal gradients, for example. Also, what's interesting here is that even though there's no clear effect of altitude, there's still a huge variation in the growth rates for the different individuals. So I think that's also quite cool, also because it's lower at 25 compared to the others. So again, showing that this 25 is maybe quite a stressful temperature for them. But the fact that there is so much variation, which is for quite a large part explained by the effects of family, for example, I think is really cool, and it also shows that there is potential for these larvae to adapt in the light of climate change. The final thing that we see here is that the males seem to grow a little bit faster than the females. Yeah, and these type of differences between the sexes are kind of known also from the literature, so I don't think that's the most interesting of these graphs. So then there's the wings, and here it needs a little bit of explanation. So we measured the wing loading, which is a measure for how much weight a wing has to carry. So for example, when the wing loading is very high, you have small wings but a large body. While if the wing loading is low, then your wings are quite big for your body size. So your wings need to carry less weight per unit of area. Here again, we see a lot of variation. We don't really see any clear effect of altitude. There is a small effect of temperature again. But I think here the most interesting one is that the males have a higher wing loading than the females. And we actually see this in the system also, that the females do the most of the dispersal. So for them, of course, it is also beneficial if they have bigger wings compared to their body mass. So to conclude, temperature affects the life history, with larvae growing faster at higher temperatures and having smaller bodies compared to their wing size at higher temperatures. There's no clear effect of altitude, but a lot of variation showing that there could be potential for evolution in the species. And also the effects that we saw of altitude point in the direction that maybe we might find something in the future here. So actually I wanted to quickly show this preliminary graph that I found on my latitudinal study, which actually shows really big differences between the three countries that I tested. So I would say stay tuned for this next year. And with that, I would like to thank you for listening, and I'm looking forward to answering your questions.